Hi, students. I want to discuss the components that make up DNA. So we're looking at 4 nucleotides predominantly. There is an exception, but I'm not going to get into that. The nucleotides that we're looking at are guanine and adenine. These are known as purines. You can see that there's a double ring structure here. There's a ring here and a ring here. And then we're looking at adenine. They're very similar, but there are enough differences at a chemical level that they produce different hydrogen bonds with their complementary base pair, which will be one of these single ring structures, cytosine and thymine. Essentially, what you need to know is that guanine forms optimal hydrogen bonds with cytosine, while adenine forms optimal hydrogen bonds with thymine. And because of that optimal hydrogen bond structure, what happens is the base pairs are able to create a structure that is stable for the entire DNA sequence. So when adenine creates hydrogen bonds with thymine, it is able to make 2 hydrogen bonds, whereas guanine is able to make 3 hydrogen bonds with cytosine. That extra hydrogen bond might not seem like it's that big of a difference because, as we've said, hydrogen bonds are weak connections, but that extra connection helps to maintain the stability of the DNA sequence. As we said, it's important to be able to have the double helix because one strand of the DNA is essentially the instructions for making a protein, and the other one essentially serves as a backup copy. This is important because if, let's say, hypothetically, a sequence was damaged, then it would be possible to repair that sequence by using the opposite strand, the backup strand, similar to how somebody might lose a copy of their keys. Well, if they have another one available, then they'll still be able to get in to their house. Having 2 strands of DNA that are mirror images of each other allows for the replication of damaged sequences if something gets damaged, but it also allows for, when new cells are made, for the DNA sequence to be replicated within the additional cells. So what we see is this idea of a DNA strand separating into 2 separate strands, and then a copy being made of the mirror image. So as you see here, the left strand is replicated here, and in its complement, the right strand, a copy is made. Whereas with the other strand, we see that the right DNA strand is maintained and a copy of the left strand is made. So now each of these strands has half of the original strand of DNA, while it has a copy of its mirror image. So what we say is that this strand has been replicated semi-conservatively. This idea of semi-conservative replication is an important idea that allows us to go and make copies upon copies upon copies upon copies, starting from the very first single cell that makes up a life. So when the egg and sperm of a life form fuse together, you start with a single cell that has the full DNA genome of that life. And from there, as new cells are made, they are made through this semi-conservative replication of the very first strand, the very first genome that the very first cell that makes up that life consisted of. So copies upon copies upon copies were made. And one way to help ensure that those copies stay consistently the same is by taking one strand of the DNA and making a copy off of that, while the other strand is also made a copy of, so that each new cell gets half of the original DNA of its parent.